We have started our live streaming, so if anyone misses on the video, you can use the link. Thank you. We would be starting in more 2 minutes. Hello, everyone. Thank you for joining today's meeting. I would like to welcome you all to today's webinar on Genomics in the Virtual Lab. Genomics in the Virtual Lab is an online program and a unique opportunity for high school students and beginners. This program is led by Professor Sona Vasudevan from Georgetown University Medical Center, Washington, D.C., BioClues Organization, India, and Nishchay, Sri Lanka, and Pine Biotech, USA. So let me start with the introduction about us. We are a US-based bioinformatics company who is working with multiple academics and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. We are doing this by providing training and tools that help students and researchers make discoveries using next-generation sequencing, data science, and other bioinformatics technologies and tools. Omicslogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the Omicslogic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical study majors, as well as faculty interested in bioinformatics, data science, and biomedical research. My name is Sonalika Ray, and I am the Omicslogic Community Manager responsible for daily interactions with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. I'm honored to have been working with a team that collectively has amazing experience in this area, including the other co-founders of the company, Dr. Alfred Tauber, who is working who comes from a medical background with expertise in oncology, immunology, and biochemistry of information. And one of these projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Elia Brodsky is the director of the Tauber Bioinformatics Research Center, and he is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics, and over the course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. These have been widely used in basic and translational research in biotechnology, pharmaceutical R&D, agriculture, and basic research. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panoff, who is an expert bioinformatician and has helped prepare many of the materials that we would use. Avi Titovsky is a computer scientist and leads the development of the T-Bioinfo platform that we would be using as a part of the course. And I will also be introducing our team members in India in the next few slides. So Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases. And she looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar, he's an expert bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. Over the past several years, he has been developing collaborations with universities as a mentor and project lead for Omicslogic training and research fellowship. Now, coming back to the program, Genomics in the virtual lab is a special opportunity to learn about genomics and big data analysis from a world-renowned scientist and a team of bioinformaticians working on cutting-edge big data challenges in biomedical research. Professor Sona Vasudevan, Professor of Biochemistry and Molecular Biology at the Georgetown University Medical Center, will present disease-associated projects that program participants can learn from to see how genomics is used at the intersection of data science and biomedicine. In this online program, participants will learn about the analysis of high-throughput genomics data and apply complex and computationally intensive data processing steps to large-scale public domain data. The program would include certification for participation as well as excellence, and the certificate of excellence would be provided to students who would exceed the progress expectations and complete the online courses and propose a novel bioinformatics project. Now, I would like to pass it on to CEO Pine Biotech, Elia Brodsky. Over to you, Elia.
Thank you very much, Sonalika. Let me share my screen. Welcome, everyone, to today's session. I'm excited to see so many of you joining. And hopefully what we will be able to provide you with is a brief view of the topics that we'll be covering, as well as introduce you to the main program coordinator, Dr. Sona Vasudevan, and some of the other participants and partners that we have joining this program to talk about their need for research in using bioinformatics and using data science. So my name is Elia Brodsky, as Sonalika already mentioned. I'm one of the co-founders of Pine Biotech. So I wanted to kind of briefly introduce what we do, why are we organizing this program, and where can you envision yourself applying bioinformatics after completing a program like this? So probably many of you have heard about the genomic revolution, right? And so the genomic revolution started in the 20th century. We're now in the 21st century, so it really started in the past century, but it didn't happen so long ago. And over the past 50 or 60 years, there has been a tremendous growth in the type of sequencing data, interest in the human genome and other type of genomic data, and really a revolution going on using such genomic data in many different aspects of biotechnology, agriculture, and biomedicine. And essentially, what genomics often boils down to is analysis of the genomic code to identify variation. And that variation could lead to these changes in the phenotype. So essentially, the change in the genomic sequence could lead to a change in the way that cells, tissues, and organisms look and function. And those could be diseases, or they could be natural variation, like different color of skin, different length of your eyelashes, or maybe even different height and things like that. So what we think of typically when we consider genomics is this idea of finding bugs in the code, right? So if those of you that are coming from an engineering background may be familiar with coding and data science, having code work perfectly is almost impossible as you're developing that code. And so oftentimes finding that bug can really help you understand what is going wrong with a function or some kind of an algorithm that you're trying to run. Now, where is genomics applied, especially in the area of biomedicine? Genomics is extremely important for understanding drug metabolism. So we understand how different proteins are able to process different types of chemicals. And typically drugs are those chemicals that we want to make sure that they're metabolized properly. You can also think of genomics in application to cancer, so somatic mutations in tumor development, inherited mutations like in sickle cell anemia, where the adaptation to different diseases has led to these phenotypic changes that are now causing some kind of a susceptibility to disease. Rare genetic diseases that could be linked to repetitive elements in our DNA, and also drug resistance. And obviously now, living in this pandemic, we know about genomics so much more because it's been in the news, it's been in the media. Everybody knows about the mutations, the variants of concern. Everybody has been hearing about this. And so it's an extremely important domain for research, but also an application to different areas in biomedicine. In fact, today, genomics is truly in the clinic. And so you can see here an example of a report that is being produced to kind of establish risk for developing certain types of conditions. And this is a routinely used method in the medical field today, at least in some of the Western countries. And so I think what's very important is that genomics is moving away from just being purely in research and being actively used in the clinical setting. And we see this as a very important development, primarily because the cost of sequencing is much lower, availability of reference data is much greater. And so there is already a clear and established relationship between specific changes in the DNA code and specific diseases and conditions that could be clinically categorized. But it hasn't always been like that. And even today, there's an active research also on the impact of specific mutations, because if you consider how these mutations occur and how do they actually lead to diseases, it's a complicated, coordinated kind of an effect of multiple factors. So here you can see the significance of DNA variation in cancer, where acquired mutations, germline mutations, mutations in different types of genes, and in DNA repair genes, all of those come together to form a certain level of risk, after which there could be an event that could trigger some of those important changes in critical genes and ultimately lead to what we know as unregulated tumor growth and development. And so these types of changes are really important for us to understand from the basics of genomics and the basics of what this data could show us about these diseases, because detecting those changes early on and also knowing the context of specific mutations can really help us understand how to treat diseases that previously were essentially untreatable. Now, more than just individual mutations, there are many different types of mutations. So there are many different types of variations, like multiple nucleotide changes at the same time, changes that accumulate in a specific region, maybe translocation, so certain regions even moving to another position, or maybe copy number variation, right? So many different types of changes in the DNA. And importantly, it's not just the human DNA, but also the DNA of many different other bacteria, viruses, and parasites that are important for the disease progression and treatment. And so I hope that in this program, we'll have an opportunity to see how genomic data could be used to understand many of these conditions, and how can we study genomic data so that we understand the important factors that are essentially critical for understanding of disease, treating disease, tracking epidemics like the epidemic that we're seeing now, or other potential epidemics that we might see in the future. But I think the critical part of this program is that it will provide you with an insight about next generation sequencing data, how that data is generated, what does this data really represent in terms of the technical part of genomic data sequencing. And then we'll allow you to evaluate some of this genomic data using these practical data sets that we will take to understand where do we see just simple variation, where can we track mutation, how do we then categorize mutations and understand what do these mutations really mean for the phenotype. And so we will make sure that in this program, after you complete the training, you will be able to understand the different terminology of what is a nucleotide, how can we categorize different changes of nucleotides, how do we then annotate those nucleotides, and how can we essentially find this association with a phenotype, including some examples of what these phenotypes might mean. Now, all of this together is really the domain of bioinformatics, because bioinformatics combines informatics, the information technologies that allow us to retrieve, store, annotate, and efficiently use different types of information, statistics, or using statistical methods to be able to analyze such data, and the domain where we apply this in the domain of life sciences or biology. So that includes biological research, biotechnology, agriculture, genetics, and biomedicine. And essentially, because of this growth of data, the availability of data, and the need to use this data in a meaningful way, we have developed this Omicslogic training that includes different types of data repositories, mentorship, and training. And this program is one example of such training that we offer to be able to equip the next generation of life science researchers, clinicians, and industry professionals that will essentially transform the way medicine is being done, the way that new drugs are being brought to market, and the way that we can innovate early discoveries that will inform different types of applications in the future. So the reason why we call this Omicslogic is because the data that we are describing here, genomic data, is just one of the omics data types that is becoming more and more useful and prominent in this field. And so Omicslogic combines different user-friendly tools, including different methods of statistical analysis and machine learning. It includes the logic behind experimental design so that we can understand the technical variation in the data. It gives you access to big data mining tools that you can easily use to study this data. And it provides also examples of how this data could be efficiently analyzed to spot significant changes in the context of real-world studies. And so this Omicslogic program is one program that is going to be available on June 21st. And it provides you with a complete roadmap to get started and learning about bioinformatics and about genomics in general. So in this program, you'll be able to find data sets, explore them and visualize them, understand research methods, and get insight and understanding, potentially leading you to an independent practice project that you can develop in a short period of time. So to kind of give you a little bit more detail about Omicslogic and beyond this program that we're discussing today, what it offers, I would just briefly ask all of you to go to our Omicslogic portal, which is learn.omicslogic.com. And so I am, sorry, posted in the wrong chat here. Okay, thank you very much. So to kind of give you a sense of how this program will work and how all of this content is going to be organized, let me briefly show you how you can get started today. So if you could go with me to our portal, and please let me know in the chat once you are there. Okay, you can put there or yes or something. You will find that there's a button to log in, and you can create a free account on here or log in with your social media. And so I want to show you briefly how this is going to work. Okay, thank you, Vasily. Manali. All right, thank you, Sonalika. So please let me know once you're in and you've registered. Punita. Okay, anyone else? If you already have an account, once you log in and you're in, you can go right here to programs. And on the program page, you will find Genomics in the Virtual Lab. And so this program right here is going to provide you with all of the information about today's session and what you will learn in this program. And after a few minutes, we will hear a little bit more about what are the different topics and how this is going to be delivered. So here you can see an overview of the schedule and some of the important tools that you will get access to. So let me briefly tell you what are some of these program resources that you will be getting access to as a part of this program. So for different parts of the program, we will leverage Omicslogic resources that are designed to help you learn about genomics. And this includes asynchronous coursework with theoretical material, quizzes, and practical assignments. Our cloud-based analytical server, where you can analyze large genomic data and visualize results. The Omicslogic code playground, where you can practice coding in R and Python, as well as a repository of project examples that will guide you from data, the logic of the analysis, to the practical exercises that you can replicate the analysis that was described in the publication. So first of all, we will have an asynchronous course that you can join, and a lot of this you can actually start looking at for free, which is an introduction to genomics. So if you're absolutely new to genomics, this is the place to start. You can go to this course to understand the applications of genomics, some of the basic terminology, maybe some refreshment of your molecular biology, so you understand what is DNA, how is it translated into proteins, and things like that. And then you will also learn about how genomic data is generated and how you can analyze this data to extract meaningful information from it. So to get started, you actually don't need to know a lot of coding. All you will need to know is the logic behind what you are trying to do. So what is the data described and how you might potentially analyze this data. And so that is going to be done using the T-Bioinfo server, which is a big data analysis platform that provides you with easy to use graphical user interface where you just push buttons and get some kind of a result immediately. And so on this platform, you will see these maps, logical maps of complex analytical pipelines that will allow you to process data from raw format to a structured format. And once that analysis is done, you will get introduced to important functions and tools using R and Python that will provide you with a clear insight about how coding is a critical component of bioinformatics and what you can do using these existing packages to quickly get results of sequence analysis and annotation. So again, we hope that this can get you started. And even before this program is launched, you can start learning about the fundamentals of genomics, of code and analysis, and the applications of bioinformatics about these kinds of biomedical applications. So now to talk a little bit more about Genomics in the Virtual Lab, which is an intensive program that is intended to provide some of the basic material and the grounding in genomics, as well as see some of the potential applications of genomics to biomedical challenges. I want to describe how these programs are going to leverage some of these asynchronous resources that I briefly spoke about and give you an opportunity to immerse yourself into this genomic data analysis experience that will combine interactive sessions on Zoom like we're having today, self-guided study materials that you will have access to, and some of these practical assignments that will include in-class assignments, but also assignments that you can do yourself, and then present your results to your peers and to the mentor of the program. So the program, as was already mentioned, is going to be run primarily by Dr. Sona Vasudevan. She has an extensive background with research, but also she loves teaching. And to talk a little bit more about herself, her background, and how this program is going to be done, I want to pass it on to Dr. Sona Vasudevan from Georgetown University. Sonalika, you probably have to make Sona a co-host for her to share.
Yes, I'm not getting the option. Elia, can you please check?
Okay, there we go.
Thank you so much.
We can
Hey,
see
there we
Sona.
are. Finally, thank you. And I'm sorry, there was some technical glitch. And that kind of leads me to what the course is all about. Thank you, Elia. And I want to thank Pine Biotechnology, Mohit, Sonalika, and others for giving me the opportunity to actually run this course. And I'm very, very excited. So good morning, good afternoon, good evening to all of you. I'm sure you're all from different parts of the world here. And it's an honor to have all of you, and thank you for taking the time to be here today to listen about what the course is all about. Elia has already done most of the work for me in terms of laying the foundation of why bioinformatics and data science is extremely important today. It almost seems like you wake up, and I'm sure all of you sleep with your phones, wake up with your phones, look up things online. So look up things on your phone and everything else, right? So technology has moved pretty fast in the last 10 years or so. I got into bioinformatics about 15 years ago, or even more, a little more than that. And I have actually witnessed every day how things have changed and where we are today. And it seems like in the last 15 years, we have huge amount of data that is accumulated. We have patient data, we have sequencing data from animals, we have so much of data. So one thing I'm sure all of you will agree with me today is that we are drowning in data from all different walks of life, right? So bioinformatics is about trying to figure out how to use this data meaningfully. And right now, it's, as Elia also explained very clearly, that the field of medicine and looking at diseases has completely changed. So we all need data science to be able to understand diseases from sequences. And so that's what I'm doing for a living. I am a professor at Georgetown University in the Department of Biochemistry, as Sonalika introduced me earlier. I also direct a master's program in the field called systems medicine. I'm not sure how many of you have actually heard about that. It's a very new field, and I call this a new language. And I feel that this language encompasses bioinformatics. And this is a language that all of us need to learn sooner or later to be able to converse and understand diseases better. So the summary of that program, or what systems medicine means, is one size doesn't fit all. So two people, you and me, may have the same cold and cough and everything, but the same medication may not work in you as well as it works in me. And we are still living in the pandemic. Hopefully we'll get out of this soon. But we all have learned one thing, that each person is different. Each person reacts to this virus differently, because we're all underlying makeup of our genome is very, very different. So how does that relate to the course, Genomics in Virtual Labs? So as Elia said, right, there are mutations and there are variations in your genome. So the way I look at things is, if there is a mutation in your gene, it gets personal. So any change in your gene is going to affect the genome in some way, and it manifests itself in diseases. I'm sure you all know that majority of the diseases, or monogenic diseases, where a single mutation affects a particular gene in the human genome that leads to diseases. There is only one mutation. So you may ask, a genome is like 6 feet, we have billions of base pairs, and how one single mutation is affecting the gene and we end up getting diseases, right? So that is what this course is all about, how important genes are to the functioning of the body, and how single mutations in a gene like sickle cell anemia or Huntington's disease or any of those diseases, how that one single mutation is altering the gene and manifests itself in diseases. So the Genomics in Virtual Lab, you all probably will see the syllabus at some point in time, or if you have not already seen that. It is 2 weeks a course, and it starts with introduction to what bioinformatics is and what big data is. And then you will learn a lot of different resources that actually are reliable resources that gives you all the information that you need to go and understand a protein sequence. Which chromosome does this protein belong to or gene belongs to? Does it have a 3-dimensional structure? And how do you relate sequence to functions, which becomes very, very important. How are drugs designed? How do we get drugs? And I'm sure in this COVID time, you all know remdesivir came about very quickly and made it to the clinic. So how did that happen? So all of those things will be covered. So we have both lecture-based and also we have hands-on that goes with it. You will learn a lot of different resources. You will learn a lot of different tools that you can apply to any disease that you're interested in working. And so that after the lecture and the hands-on sessions, and we'll also have a renowned person in the field of bioinformatics that we will invite, who will actually give you the importance of bioinformatics from NIH or Georgetown University or George Washington University, and who is going to tell you the importance of bioinformatics. So from a practical aspects, by the end of the course, you're all going to know where bioinformatics started and why we cannot live without bioinformatics in this age, and what is the relevance of bioinformatics in medicine, and how do you understand diseases, and in fact, what causes those diseases is something that you will actually learn. It's a very intense program, but hopefully we'll make it fun. I like fun. I'm not a serious lecturer, so it'll be fun. We'll have games. We will do lots of things. We will go at your pace, and finally you will be amazed at the project that you're going to come up with. And so I'm hoping that it'll be very fruitful, and you will learn a lot. And Pine Biotech, as Elia showed, Omicslogic is an outstanding platform. In my opinion, it's one of the best platforms that we have that encompasses so many different aspects. So you're going to become budding experts in bioinformatics. And from practical aspects, it's going to pay you a lot of money in terms of jobs. So job opportunities are huge for bioinformatics right now. And most of my students who just graduated from my program already have jobs. So from a practical aspects, this is a skill set that's going to be very, very valuable to whatever you want to pursue in your lives. So with that, I will transfer it back to Elia, unless if you have any questions, or if Elia, you want me to talk about something else or cover or any other aspects that you think that you want me to talk about.
Well, no, first of all, thank you for this overview. I do want to ask if there are any questions from the participants today. If you have any questions for Dr. Vasudevan, you can post them in the chat, and she will have time to answer those in just a few minutes. Also, Sonalika, I wanted to ask that you share the link to the form for those that might be joining us from places that we want to discover. Sonalika? Okay, thank you. So as you're thinking about your questions, this is a form that will help you understand better. Those of you that have joined today, we are asking that you fill this out if you would like to receive future information about the program start. The first session of this program is going to be open also to everyone. So you're welcome to invite your friends and contacts to join the first session, which will be discussed. The whole schedule is going to be discussed at the end of today's session. And again, if you do have any questions, feel free to post them in the chat, and we will have a time for Q&A in just a few minutes. Now I would like to pass it on. Sonalika, maybe you could introduce the next speaker.
Thank you so much, Elia. Now I would like to invite Dr. Sugunakar Vure from BioClues. Dr. Sugunakar Vure is presently Assistant Professor in Lovely Professional University, India, and also he is the Secretary of BioClues Organization, India. He has been earlier postdoctoral fellow in ICMR and also Chungnam National University, South Korea. He has done his PhD in cancer genetics and is an expert in the same field. Over to you, Dr. Sugunakar.
Yeah, thank you so much. I hope I'm audible to you all.
Yes, sir, you are audible.
Yeah, Aparna, could you please share the screen? Aparna, are you on? Yeah, please excuse me for a couple of minutes. Aparna? Yeah.
Yeah, 1 second. Aparna, yeah, we need to make her a co-host. So just one moment. Okay, there we go. I think it should be working for you now.
Yeah, thank you, Aparna. Yeah, it's pleasure to be part of this evening. Thank you, Elia. Thank you, Sona Vasudevan. All the participants. I take this opportunity to just brief about the BioClues organization, where I take up a position of as secretary. Basically, BioClues has started in the year 2005 with an idea of bioinformatics club offered by experimental scientists, and the BioClues has got a mission under mentoring, outreach, research through awareness, and entrepreneurship. So further, when we talk about BioClues, it's spread across different countries, more than 40 to 50 countries, which we are currently working on. So talking from India, Australia, US, and even we have started uh, other places across the globe. And the milestones, we talk about way back in 2005, where the virtual organization was founded, co-founded by Dr. Prashant Surabarjula and Pratish Vardharaj. And in 2016, first virtual projects conference took off. In 2007, first publication and several conferences been organized. And it's been 16 years now, where we have covered under various uh, points mentioned over here. In the 2009, I'll just try to give some glimpses. Where 2009 we started Women in Biology Forum, started with full-fledged society, and then we become the ISAB affiliate, introduced the Bird Awards, which are specifically given to the people who are has got expertise in the field of bioinformatics. So to encourage in the Indian scientific fraternity, and also recognize people abroad. And in 2012, we have incubated IKP Knowledge Park at Genome Valley, India, started an open access journal, peer-reviewed journal, Journal of Computational Biology, and first ever toll-free mentoring in bioinformatics. In 2013, BioClues Overseas PhD program, a joint program with Aalborg University, Denmark. And 2014 to 2019, publications, we basically follow publication co-ethics like life memberships and travel grants. in 2009, we joined forces with Pine Bio towards education programs. And then it started in the year 2020, we started with bioinformatics for schools. And then we also started international chapters. And regarding sustenance, we use inverted paid memberships, corporate memberships, virtual on-site conferences, project resources, and we do grants and funding. Yeah, please, next slide. And to give a brief introduction, like founded by Dr. Prashant Surabarjula and Pritish Udiraj, as I told, and the present president is Narahar Shastri. He is the director of CSR NEAST, uh, and the immediate past president, Dr. Jairam Valadi, and the honorary president, Dr. GPS Raghav, and several other vice presidents. And it is being registered as not for profit organization and adheres Creative Commons license. And there are 4 avenues, as I mentioned: mentoring, outreach, research, and entrepreneurship. And approximately we have around 4,500 plus members right now, and across 40 countries it's been spread. And currently we do multiple projects, and more than 250 scientists are involved in various projects, and a lot of collaborations that come across different 10 universities and organizations across 8 countries, and around 40 publications and over 100 conferences have been presentations have been given. And we published in Impact Finder Journal between 1.15 to 11.836, and we do conduct conducted 5 virtual conferences and several international meetings organized, and only official direct affiliate of International Society of Computational Biology and Asian Specific Bioinformatics Network in India. Yeah. Mm. I think Aparna, you can go ahead from this slide. Yeah, thank you.
Sonalika, may I start?
Thank you so much, sir. So now I would want Dr. Aparna Banerjee. Dr. Aparna Banerjee is Assistant Professor in Catholic University of Malaysia and also the present Joint Secretary in BioClues Organization, India. She has done her PhD from the University of Burdwan, India, and her area of expertise is in applied microbiology of extreme environments, comparative genomics, and bioinformatics. Over to you, ma'am. Thank you so much.
Thank you, Sonalika. Thank you, Elia. Thank you, Dr. Sona Vasudevan and Dr. Suguna Karvuri. So I will take the next lead to talk about BioClues. BioClues have, to system biology, have done a lot of system biology workshops in virtual mode occasionally too. So there have been 3-week bioinformatics workshops scattering the needs of scientists and the wet lab professionals. More than 1,500 people benefited of the organization that we have made of nearly 49 such events. Eminent faculties from India and abroad joined over there. Several workshops have been organized in Colombo, Sri Lanka, and all over the India. We have rigorous teaching program project in collaboration with several organizations like National University of Singapore, Florida University, United States, and in Italy. We also do certificate and honorarium at the end of the workshops. Sonalika, I cannot move the...
Ma'am, maybe you can reshare your screen. That would work, I think.
Okay.
Stop sharing and then reshare.
Okay. Yes, so as a resource, BioClues organization have competent core team. We have prolific workshops and several events. We have Blade server. We go through mentor-mentee relationship, as Dr. Buri also mentioned earlier, and we have several BioClues interest groups. So some notable projects of BioClues has been Cancer Institute Chennai, accessing the human smoker population prone to lung cancer, a checkerboard analysis using NGS data. It has been also with Ludhiana University, a bacteriophage genome, Brucella fudge, annotation, and there has been also the HYPO project, prediction of hypothetical protein function in humans. About the NGS expertise, the studied organism has been human cow yeast bacteria through the platform of Illumina, Roche, parent, and multiplexed. The services that we offered are mentoring graduate students in biology and bioinformatics, annotate and curate NGS datasets, transcriptomic and genomic analysis of NGS data that includes gene expression analysis, variant analysis with or without reference genome, genome diversity and coverage distribution, pathway analysis and protein interactions, functional mapping with gene ontology and pipeline developments. So talking about the gene, our team, Prashant Surabjalla and Pritish Bharadwaj, they have co-founded the organization 16 years back. Now the code is formed with President Narahari Sastry, the secretary is Sugunakar Bhuri, with me being the joint secretary. The finance chair are Harpreet Singh and Santoshi Dupatla, with the women in biology chair with Sakshi Singh and Renuka Surabjalla. The student chapters are done by Arvind Preet Kaur and Rakshmi Sukumaran. The internship chairs are V.S. Sundararajan and Anil Kumar. The outreach chair are by Jairaman Valadi and Mohan Kumar Megha. The research chairs are by P.V. Kavi Kishore and Hima Kumari, with the mentoring chairs finally with Biswanadam Dupatla and Prashant Surabjalla. Many of them actually today has joined with us, Prashant, Dr. Sundararajan, and they have all been the driving force to BioClues. Here are the mentioned honorary advisors of BioClues 2 with the founding board or executive members. So the latest conference that have been successfully organized was INVIX 2020, with being the affiliates of ISCB and APB Bionet. So there are some glimpses of INVIX 2020 workshops that we organized, 3 workshops, and then some glimpses of INVIX 2020 here. And finally, we have also started in 2020 Bioinformatics for School Children program, and 2 batches have already graduated. The 3rd batch will start soon also. So we focus on hands-on and home-based learning, and we emphasize on strong STEM and curriculum product development with a total of 12 intensive classes. So here we thank you all for giving an opportunity to talk about BioClues, and we will be happily sharing our expertise in this Genomics for Virtual Lab program. Thank you, Sonalika, and thank you, Elia.
Thank you very much. And I want to just now pause, and I see that there are several different questions. Maybe we could address those questions now. So first of all, there's a question, and Dr. Vasudevan, if you could please answer these questions. So Hira Iqbal is asking, for a basic science person, what should be the first step to land in the vast ocean of bioinformatics?
Actually, Hira, that's actually an outstanding question, because you know what, I had to swim in the ocean. I don't know swimming. So now having gone through a long process, I can tell you the first thing is to actually take this course, because that gives you a foundation of bioinformatics. And if not this course, because it may seem like I'm advertising for my own course, but Pine Biotechnology actually has lots of introductory courses in Genomics 1, Genomics 2. I think those will be a good start for someone from basic sciences. And I am actually trained as a basic scientist myself. In fact, I'm trained as a physicist, and then moved to biology, and I'm from your same background as basic sciences. And I think when I was trying to get into the field, there were not that many courses or any of these things. So it was a hard way. So long story short, it's a long answer for a short question. I think any of the introductory courses that's on Pine Biotech will be a good start. And of course, we'll be very happy to guide you further if you need more information. And then the second question is, for a biochemist, how can we relate pharmacogenomics with bioinformatics? Actually, that is actually a very relevant question to the times we are living in with respect to pharmacogenomics, right? So pharmacogenomics is the other term for personalized medicine, precision medicine, or systems medicine. And so how can we relate? So for pharmacogenomics, in my opinion, and I'm sure Elia can add to that, is you need foundations of structural biology. Because at the end of the day, you want to learn how to design drugs. And then once you know how to design drugs, we can actually use all the approved drugs that we have today to do some repurposing. And so in my opinion, it is, and biochemistry is actually fundamental and prerequisite to most of the biological processes, because, you know, once you have a gene, you know how biochemically it is actually working in the cell. Added to that, if you add the omics part to it, I think that will be very, very important. I'm not sure if Elia, you want to add something to it.
I just wanted to ask you, I think your background is in structural biology, right?
That's right, that's right. So just to give my path to where I am today, I actually started as a physicist, and then by accident, and I'm trained as an X-ray crystallographer. So I did a PhD in X-ray crystallography, and then my postdoctoral training was actually in wet lab molecular biology. That was my first introduction to biology. And then, so now I'm able to, then I entered bioinformatics, and now I'm able to look back and combine structural biology, because that is helping me with respect to understanding pharmacogenomics and how one drug works and one doesn't work. It's all at the end of the day. We look at biology in 3 dimensions. And so I think that's useful.
And also maybe we can mention that, you know, as a biochemist, you know, not only can you relate pharmacogenomics and bioinformatics, you can actually participate, because I think a lot of people that don't have that biochemistry background, they don't appreciate as much the interaction between the structures. You know, they might be only looking at the sequence level. But in this program, I think Dr. Vasudevan will mention the importance of keeping those 2 things in mind.
That's right. That's right. So it'll be a natural transition from first principles biochemistry, amino acids and how they fold together and how the structure is made and how the structure define the function. So thanks, Elia, for that note. So let's see.
There is a question, what's the importance of Linux platforms in bioinformatics, and do I need to learn Unix for further studies in bioinformatics? From Sulahuddin.
I think, as Elia said in his introductory, I think there was a very nice slide where I like the slide how you define bioinformatics. So bioinformatics actually has different wings. So you are a hardcore biologist, or you are a software engineer, or you are a biostatistician. So they all come together, because bioinformatics is a very interdisciplinary field. So it depends on which wing you want to pick out. So I am a hardcore researcher. So I have programmers who do the programming part for me, and I don't do programming personally myself. So it is an interdisciplinary field. So whichever wing you want to choose, whether you want to choose research wing, where you use other people to do the programming, or you can be that software engineer to actually use. So which means Linux platform is actually very, very important in terms of most of the tools right now. But Unix, Elia, do you want to?
Yeah, I would just briefly add to that you know, there are many limitations to user interfaces. A simple example, if you want to open a large table, you can only open a table that big. And so because these user interfaces want to load everything into the RAM, the more efficient way to do that is to use Linux that has access directly to files without necessarily loading all the files. So you can extract information out of a file without opening the whole thing. As we move into the field of big data analysis, that's critical, because every file is gigabytes and gigabytes. And so you just can't operate that without huge resources. So it's a more efficient way of doing things. The other part of it is that many tools are designed around Linux. And so simple settings of those tools, how to run those scripts, how to combine scripts into pipelines, all of that typically is done on Linux. But as Sona mentioned, there are many variations of bioinformaticians. And so there are people that design the algorithms, use the algorithms, interpret the algorithms, outputs, but even generate the data for those algorithms to use. All of those people, I think, need to use bioinformatics at a certain level. And so for this program, we're going to be focused on kind of user-friendly bioinformatics, things that you can do on Windows, on Mac systems. You don't necessarily need to know Linux to get all the tools that we're going to be using here. But what you will be able to do is think critically about the data and the application of these tools to potentially research or biomedical challenges.
So there's a question about from Manali. So you graduated from bioinformatics and you want to do a master's in and eventually you want to do a PhD in bioinformatics, which is fantastic. We really need more trained people to help us all here. But my personal advice to you, I'm sure other people would can actually add to this. My personal journey has been that trained as a structural biologist, I actually did biology in the lab. And so I find that I'm able to appreciate more of the biology side of things because I've been in a lab. If I had completely done only sequence analysis just using a computer, I don't think I'll be able to appreciate the biology of it as much as I am now able to understand it better. This is my personal view, that my advice to you would be to pick a PhD where you actually do lab work so that you can actually come up with predictions using bioinformatics, but actually authenticate that prediction and validate the predictions in an experimental setting that makes you a full person to understand different things. That will be my advice. But of course, there are other experts in this call. So if someone else wants to add to it, please do.
And Sona, could I just ask, I think that a lot of people are interested in joining a master's degree program. So I know that you're a director of a master's program. Could you tell a little bit about the program itself and what kinds of qualities do you look for in students that are applying?
Yeah, thank you so much, Elia, for giving me the opportunity to talk about the program. So Georgetown has the master's program. Maybe I can actually open up the website and I'm not sure if I can share. So, so this is the master's program and it's a 10-month program. It is from start to finish. And it actually trains you to all different aspects of bioinformatics. So it is like a layered approach. So what background you can come from, any background that is computer science or biology or bioinformatics or any discipline, you can enter the program. And what the program, when you leave the program, you're going to have a full flavor from start to finish, different layers of informatics. You'll learn about genomics, you'll learn about proteomics, you'll learn about microbiome, and you will learn about different aspects of how clinicians are using bioinformatics. And then you'll also learn how to critically read scientific literature and how do you translate all of this information from all of this data in a clinical setting. From bench to bedside, if I may say that. And then we'll have clinical metabolomics. And so this is a layered approach. And also the most important part of this program is you also get to do research. In fact, this program, that's how I got to interact with Pine Biotech, and they play a significant role as well in helping with different courses and as well as different aspects of the program as well. In fact, there's one course that's machine learning that Elia actually ran, and I want to bring that course back. And so this is different aspects layered, and you learn about statistics. Whenever you talk about data, whenever you talk about big data, you don't know whether it's statistically significant. And we are talking about one mutation or one variant that we are seeing. is that variant statistically significant, right? So now we're talking about lots of COVID variants. How many people have been sequenced? How many people actually have that variant? Is it even significant, right? So statistics is a big part of it. So this is a program that you're all welcome to apply. The deadline is actually July 1st. I'm not sure how many of you are ready to apply, but I know most of you are probably in high school still, but this is a program to consider or, spread the word around. We still have a couple of spots left for this coming fall. It starts in September.
So I posted the link to the site in the chat, anyone
Yes.
that is interested. And I do want to mention that even if you scroll through, you can see genomics in the lab. And
That's
so
right.
this experience is kind of like a view into what this program is going to be all about. And obviously you'll have access to Dr. Vasudevan here, and you can ask her questions about any of the topics that you might want to learn in preparation for this program.
Yeah, so Genomics is lab, that is pretty much where, this is literally in the lab. So since we're not going to be physically in the lab, so this is what we call it the virtual lab. So pretty much we're doing all the experiments in the computer instead of the lab. Thank you, Elia, for giving me the opportunity to
all right.
the program. So
thank you, Sona. So I think that now we heard about the program, the topics, and I think it will give you a view of what a bioinformatician really does and how they can apply different bioinformatics tools to a variety of different challenges. Now to transition, I want to pass this on to Dr. Mohit Mazumdar, who will guide us through the schedule of the program and the registration.
Thank you, Elia. And hello, everyone. Let me know if you can see my screen.
Yes, sir, your screen is visible.
Is it visible?
Yes, sir, it's visible.
Sorry, maybe I'm not hearing you back.
So your screen is visible.
No, yeah, now it's okay. Thank you. Thank you so much. Yeah, hello, everyone. And thank you so much, Elia and Dr. Professor Sona and Sonalika to help and all the BioClues members to help us guide through the entire program and how it will be, what are the benefits, what are the applications, whom this is for. So genomics in the virtual lab, and this is the second version of it. So we did genomics in the virtual lab in 2020, and we have had like really amazing results working with many high school students, and also undergraduate and postgraduate students from India joined, and many other places who would want to get understanding of how genomics is applied in different areas of bioinformatics and different areas of life science and biological research. So one thing that I think we have discussed quite a bit is the application of genomic research in biomedical research, in translational biomedical research. But with our collaborators and the phases of this program, which I'll be going through right now with you, all of you, we will learn that you will learn that genomics can be applied to a various different areas like agriculture, and many of you might have a bent towards working on something like that. So we'll be able to help you that during the project phase as well. So now talking about the program, so it has the entire program is divided into thinking of a concept of how genomics can be applied in basic research, and as well as how genomics can be applied to understand healthcare research and healthcare system and improve the healthcare system. So from developing a hypothesis to be able to work on translational research. So those are some of the objectives of this program. So introducing the program from the first week, we will have the introduction to bioinformatics session. So that will be the first session, and as Elia already have said, that this session will be freely available to those who are interested in joining the entire program. So in this session, we will start with the basic of bioinformatics, understand how bioinformatics started, understand its application, and we'll go through a coursework of Introduction to Bioinformatics, which is available on the platform that you will have access to. So followed by that, the second day, we will be learning about protein structure and function. This will be again with the help of Dr. Sona here, Professor Sona here. She'll be leading this session and letting us learn about how protein structure and functions are related. And then we will learn about sequence analysis and sequence alignment and phylogenetic analysis, and how these evolutionary analysis helps in evolutionary and understanding of evolutionary distances, and then the research that enables us to learn about the origin and understand all those questions that are relevant to today's biomedical problems. So next session, we will have about mutations and 3D structures. And by this time, we will learn about how we can apply some of these techniques on different data sets. So we started to work on data sets, and then we have a review and activity session. And this will be a guest session where we'll have games and we'll have different kind of activities that will be associated with the with aspects of genomics. And the guest lecture will involve someone, which is again a surprise, and which we will be announcing very soon. Will give you an insight of how genomics is being applied, and is helping a lot of people with many disabilities and problems. So after the completion of the first week, and that will be by 25th June, we will be finishing the last session. And during the weekend, we'll have weekend activities. And during this time, before the end of the week, we will be providing you with the details of the assignments, and how you can complete the assignment, and what all other elements are there that you need to complete. And we will be sharing those details with you, the data sets. And then we will have a review session starting next week, where we will be reviewing the video presentations. And we already have a lot of examples of people completing these weekend activities. And I would request Sonalika, if you can paste some of the links of the participants who completed the program last time and completed the weekend activity videos, so that so the students here can understand that how it could be. So the second week is going to be more about research. So you understand about the structure and function, how genomics is applied, and with the help of Professor Sona, we will have such amazing interactive sessions that I can assure and guarantee you that because last time we have had an amazing experience going through this entire journey, learning from her, learning from different tools that she's being part of while developing. So after that, we will be transitioning into the second week, where we will learn about the application of genomics in virology. And also we will understand from many case studies and how you can apply this understanding of genomics by first doing literature review. So we will start from the basic of research, and we have a very structured programs that are leading to high value research. So we have several examples of those, and many of our interns who have joined are working on research projects using the same process. So we start from literature review. We will go through different repositories that you will learn and you will find interesting articles, and that will be really an amazing experience for those who are interested in doing something new, a new research. So finding out what has been done and how people have achieved those will be some of the objectives of this session, and how you can design a process and design a project using that information with the help of experts. So this will be the first session after the review of the first week's activity. So review process and feedback process is a very important pillar in this Omicslogic bioinformatics research trainings, and especially genomics in the lab. We have sessions, very intensive sessions, because we have sessions every day. So we'll be expecting you to be indulged with all the experts and make the most of this opportunity. So next session, after learning about and identifying the research question, we will be learning about how you can, identify viral species and strains and do comparative analysis of viral genomes, understand the viral diversity, and then understand how this is related, how the mutations are related to the disease, how it is related to the disease pathology, what are the phenotypic to genotypic changes. So how machine learning and how next generation sequencing is playing a key role in genomics for virology. So that would be the second session of the second week. After that, we will learn more about the diversity of microbial communities. So here we will learn about bacterial genomes, and then we'll learn about 16S RNAs, and we'll learn about how metagenomics can be applied to study directly samples that you can culture in the lab. So that is such a big advantage that we have with the metagenomics technology, and you'll be learning about how metagenomics technology can be applied to answer many of those important questions in this difficult pandemic time where there is so much of data is being generated related to COVID-19. So after that, once you have applied and learned about how genomics, metagenomics plays a role in identifying, understanding in the respect of finding these problems that you're working on the aspect of project and all. So in this session, we will learn about the basic principles behind antiviral and antibacterial drug and the approaches for vaccine design. So it's a very important topic, and this can be done like once you have the holistic understanding and overview of all of the topics that we covered, we come to this stage where we are talking about antibodies and antiviral drugs. So the earlier understanding is equally important for you to come to this stage and to make the most and understand the translational value of genomics. So the next session after that on 2nd July will be about hands-on session analysis of genomics from NCBI virus. So we will be going to NCBI virus. We will be downloading the genomes and we'll be using the T-bioinfo server to analyze those genomes. So we also have some of the project examples that we have already prepared for you for this program that you will be utilizing and learning from. So those examples will also be available on the Learn Omicslogic platform that you will have access to and subscription to you throughout this program. So after the completion of second week, we will be working on the weekend to develop these projects ideas into presentations. So this is a first step to work towards a program. So one of the practical outcomes of this program is that you get a feedback from a renowned expert in the field of genomics, in the field of disease research, in the field of all the other genomic application. So we have renowned scientists from all around India, all around from many renowned universities in the US. So you will have an expert panel to discuss about those project proposals that you will come up with. So one of the objectives, as I was telling you, is to develop an independent research project that could be reviewed in this entire process. So after the completion of the 2 scheduled, weeks, so the first phase is the first week by Dr. Sona, the second phase by Omicslogic team on all of this that you have seen, and the third and the fourth week will be more about research project. So we will be working with experts and we will have group sessions and one-on-one sessions to develop a research project, and that requires a few steps. So I'll just quickly describe you those steps. So the first step to a research project is to find a problem, right? First you have to find a problem, and then you have to be very specific and be focused that the problem that you're interested in. So that would be from you. You will have that opportunity to come up with your own research project, and also will have opportunity to work on already prepared projects that are available. So after doing that, we will be doing the literature review, as I've already told you, and then we will do exploratory analysis where we will look into different data sets and find those insights from that data set. So entire process of working from week to week will help us develop some of those research projects. And I think I'll also request Sonalika to maybe share some of those completed research projects with you here as well. So this is about the program and how we are going to go through this journey, 1 month journey, research journey. And now I would like to maybe take you to the registration process and show you how you can register for the program. So maybe I'll find the link in the chat. So Sonalika, if you can paste the registration. Okay, it's here. It's the blog post. So once you have a login, you will find yourself like this, like a name. And then in this, you'll have a profile. So once you log in, you'll see like this. But if you have not logged in, then I recommend you to create a free login on the Learn platform, on the Learn Omicslogic, and start exploring many of the opportunities that we bring you here. So Genomics in the virtual lab, you can find it here. And then you go in and then you start your application. So this is a free workshop, and if you want us to send you more emails and details about the program, then you can request for more information and submit your details. And as you have joined this program, I believe many of you have already registered. So you have done your registration part, but now you have to finish your registration by completing and submitting the registration fees. So I'll go to the registration fees and I'll describe you different levels of levels that we have for you to join and explore. So beginners level, as I showed you, all of those things that we will do has all the features to complete this course and get a certificate. And then we have the intermediate level. If you think that you need additional sessions with one of us, one of the mentors, then you can opt for the intermediate level. So that will give you an option to have a couple of sessions with the mentor to guide you and give you an hands-on experience by doing screen sharing and helping you go through the resources and all of that and the tutorials. So if you require that level of support, you can opt for intermediate. And the third option is advanced. Advanced is for an additional work additionally on your project. So if you are working on a project and if you already have a proposal ready and you're working maybe with a lab, you are working with some university, so you can opt for an advanced license as well. So once you select a license, and I'll go here, then you have an option where you can fill up about the details, your card details and how you're going to submit the fee. And at the same time, you can also add promotional codes. So these promotional codes are given up as scholarships. So we have given a lot of scholarships to our previous participants before who would want to join this program and could not afford the full price of this Omicslogic program. So you can apply for scholarship, and I'll request Sonalika again. I'm so sorry that I have to request you again and again, but please, if you can share the scholarship form as well, that would be great. So yeah, as I was telling you that with scholarships, you can again do this. And if you're a BioClues member, then please reach out to BioClues members who are available here, and maybe any of you can share the email address to reach out to, and then you will get a promotional code from BioClues, and then you can apply that BioClues promotion code and get scholarship from BioClues as well. So I welcome you all to complete this and join us for this 1 month hands-on mentor guided research training program. And if you have any questions and any issues, feel free to reach out to us and we'll be happy to help you out with this. So my email address is mohit@pine.bio, and I think we'll share other email addresses as well. So thank you. I think I'll now look at the questions and I'll pass it on to Sonalika for any other questions that you may have.
Thank you so much, Dr. Mohit. Here in the chat, I have also shared the blog post link with you all so that you can go through what all would be covered. All the session details are mentioned there as well. And also there are few of the sample projects by some of our students from the previous Genomics in the Virtual Lab program. And also I've shared the links of those projects so that you can go watch them and get the insights of how it is done, what the outcomes are, and how things would work actually. And also I've shared the scholarship form. So the process is, once you fill out the scholarship form, you have to submit your CV and also the references from your institution. We would be reviewing them and then we would get back to you on that. And as sir has already mentioned, the registration form, I'm also sharing the link with you. Those who are interested, please register. So once we get your information, we would get back to you soon on that. And if you have any doubts, please mail us at mohit@pine.bio or marketing@pine.bio. And before we end this session, I would welcome you all to follow us on LinkedIn as well. Following us there and being a part of the group would help you know about all our upcoming programs and trainings, and also know about whatever courses we update. It was nice to meet you all here. Thanks for being a part of the webinar. If you have any doubts, you can reach out to us. And thank you so much, BioClues team. Thank you so much, Dr. Sona Vasudevan, for coming and sharing such mind-boggling things. And it was a pleasure to have you here. Thank you so much, BioClues team, and thank you everyone for being here. Now we'll meet in our next session. Let's all meet there and discuss more about the program and begin with the program on 21st. And even now, I am myself very, very much excited to be a part of the program. Thank you so much. We'll see you all soon in the sessions then. Bye-bye. Take care.